Welcome back, everyone. We are now into part 2 of the Unit 10 lecture, where we're going to be dealing with acids and bases, alkalosis, acidosis, and so forth. The main thing here with this slide is that when we have intracellular management of pH, the two systems that we use in intracellular are phosphate, notably this HPO4 2-. It can protonate or deprotonate. It's a phosphate buffer. The key is to recognize it as phosphate. And secondly, we do have side groups in amino acids that are acidic and basic. If they're weak acids or weak bases, they can function as mild buffers in the intracellular environment. Will also have effect on protein shape, but we're not going to worry about that too much. All right. The main point here is that the two systems intracellularly, and we're just repeating ourselves here, phosphate, particularly this little ion right here, this HPO4 2-, and or the side groups in amino acids, whether they're linked into a larger protein or they're just amino acids floating around inside the cells, are what help control pH inside cells. And intracellular is actually a little more acidic than blood pH. There's a lot going on in this slide, so let's try to make the message as simple as we can make it. In addition to buffering acids and bases, we may have the need to eliminate those acids and bases from the body because it's a moving sidewalk. We're going to keep making more. So one of the ways, in addition to buffering acids and bases, is elimination. So how do we eliminate things, acids and bases from the body? Well, we have a respiratory component, and that's where we can, things that are volatile, that are able to evaporate readily, can be breathed away through exhalation and ventilation. CO2 is the big one. So let's just try for a second. We breathe out. 2/3 of that CO2 just seconds ago was actually in the form of a molecule known as bicarbonate. So when we convert bicarbonate into CO2, the other product is water. So water is great for us, generally speaking. And if we get rid of that CO2, then we have a way of basically eliminating acid by converting it to water. And that's what we call volatile acids. They can be eliminated from the lungs. Kidneys, on the other hand, can eliminate what we call fixed acids. These are acids that cannot be converted into a gas or a breathable thing. Ketones are a little bit of an exception, but we'll just let that go for now. These other ones, phosphoric acid, uric acid, lactic acid, and many ketones, not all of them, because if you have ketoacidosis, you will have breath that resembles like you've been chewing a fruity gum. It's really extraordinary what the smell is, because those are volatile acids, but a lot of ketones are fixed. And so we have to use kidney mechanisms in order to eliminate these acids. And how do we do that? All right. Or alkaline substances. It's the same story, but we can start with acids. That's fine. We can either absorb or create bicarbonate. It's a lovely molecule that can absorb protons and become carbonic acid, and we can convert it to CO2 and water. Or we can even give up a proton if we're really in trouble. Okay, so we can conserve and resorb bicarbonate. We can also directly secrete protons into the filtrate, through nitrogenous situations and so forth. I'm not worried about the cells. That's a little too much. Come on, man. We can also excrete bicarbonate. We can also do that. Again, these things are sort of responsive. Your brain and your kidneys will sense these things and secrete hormones in order to trigger these behaviors. So these are a little slower. Breathing is second to second. All right. Kidneys are a little bit delayed, hours and sometimes days. So if we generate or create or resorb one of these, it's the same as if we can get rid, as losing one. Urine tends to be a little acidic because we're actually more often than not secreting fixed acids into the urine. So I believe these next few slides to be completely inappropriate for a 100 level class. I'm just going to say that. It's showing you some mechanisms and we've got transport, but we don't know enough about cell biology at this level to really understand this all that well. So what we do is we often will bring in HCO that will then couple it to a lot of processes in the body are coupled. And so we are to get rid of protons, we will resorb bicarbonate. That's all this is saying. Same thing here. We can create new bicarbonate as well. All right, that's another possibility. We can either resorb bicarbonate or we can create new bicarbonate. This is where the phosphate and so forth is linked. Again, I'm moving on. Just way too complicated. We can also create bicarbonate through certain types of ammonia and amino acid metabolism is what this is saying. This is why we see a lot of ammonia and a lot of nitrogenous waste. So this is getting rid of nitrogenous waste that help eliminate excess acid. We can also metabolize amino acids in this same thing. That's all this is saying. Again, way too complicated. So those last three slides should really just be a single simple message. Don't get into the details of the mechanism. I'm not going to test you on it. I just don't think it's appropriate. Even graduate level physiology, you're just beginning to touch on some of that stuff. And ask me how I know. Okay. In any case, this one, on the other hand, this one is real, and this one you should be paying attention to. This is a simple way of assessing what might be going wrong with the patient. And so I think this is good for you guys. The question is, okay, are they acidotic or alkalotic? Are they less than 7.35? Are they more than 7.45? Acidotic, alkalotic. Is there a problem? So if we're acidotic, it could be either because CO2 is high or perhaps bicarbonate is low. If it's alkalosis, it could be CO2 is low or bicarbonate is high. They tend to flip-flop, not always, not always. Okay, I'm sorry about that. It's blame the guy upstairs for this, but we can make a normal, call it normal, assumption that these things tend to flip-flop. So if this one's high, this one tends to be low. If this one is low, it tends to be high. And we can kind of go there. All right. Which readings fit the change in the pH? Do we have low bicarb? Do we have high CO2? Do we have low CO2 or high bicarb? That's the steps here. If it's high with CO2, that's respiratory acidosis. Either they're not breathing enough or there's something going on here that they're not eliminating CO2 properly. COPD, for instance. If you have a problem with bicarb, that could be a kidney problem. You're not either resorbing enough bicarb or creating it. All right, that's metabolic. It's just that simple. This will strike fear in the hearts of many a nursing student or many a pre-medical student. Again, these things tend to flip-flop. If this is high, this one will be a little bit low, but if the predominant reading is high, then it's respiratory. Predominant reading is low, it's metabolic. Same story with alkalosis. If the predominant reading of CO2 is low, that means a respiratory component. If the predominant reading is HCO3 is high, that's a metabolic component. So learn how to figure out what is the problem. Are they acidotic or alkalotic? And then is it respiratory issue or is it a metabolic issue? So two main questions. Okay. Rome. Respiratory opposite metabolic equivalent. All right. Whether it's CO2 or bicarbonate. And this is the big one. This is the monster in the room. There are other ones, obviously, but this is the quick and easy one, relatively speaking. And now this slide desperately tries to show you how to do it, and I think falls flat on its face. Okay. What they're trying to do is to do some sort of this quasi, it's not math, It's not. Okay. The question is the ratio of bicarbonate to CO. Oh, what? Okay, fine. All right. So what do we have going on here? You're looking to see if, again, if their pH is high or low, if their CO2 is high or low, or if their bicarbonate is high or low. My mother-in-law is on home oxygen, having been a most of her lifelong smoker, and so now she's reaping the consequences of that. And one of the things that happens was that her oxygen was a little too high, and since her CO2 drive is generally knocked out, she literally stopped breathing. And so what happened was she developed a type of acidosis secondary to that breathing stop. So this does become somewhat important down the line, but again, I think this is a little bit too much for us. So we're going to kind of just let this go. The key is to look at the normal values. If you have a lot more bicarb than what should be normal, or if you have a lot more CO2 than what should be normal, you can begin trying to decide how to manage that. At our point in time, this is probably just a bit too much. So the next set of slides is from a different PowerPoint presentation. Some of it is repetitive to what we've already talked about, but I think it helps us to at least look at the slides, talk about them, to decide, and again, see the points of emphasis. Your book is separating these things into two different chapters, but they are intimately linked, and in some cases, books will unify these chapters. But let's go ahead and move through these slides. We should already know this, that water we regulate, all right? Electrolytes we regulate, acids and bases we regulate. We've got the fluid compartments, right? All right. Intracellular, extracellular, and then the blood plasma versus interstitial fluid. We have to distribute them properly, and a lot of the ways that we move water is through the osmotic component. That water will move down its concentration gradient into higher concentrations of dissolved electrolytes and solutes. Probably should have put this slide earlier, and in future iterations of this course, I might just do that. What's the difference between an acid and a base? Okay. An acid is something that can contribute a proton to solution, like for instance, carbonic acid here, which can form just by carbon dioxide and water reacting together. So we get carbonic acid, and if we're not at the right pH, it will donate a proton to the solution, and that's what contributes to pH. So if we are mildly acidotic or alkalotic, if we're mildly alkalotic, what will happen is any carbonic acid in solution will donate a proton in order to create more acid. A base, on the other hand, is a molecule that can receive or accept a proton. For instance, here, NaOH is a base. That's because when it dissolves, it forms these OHs. They love to react with protons to create water. Thus, will reduce the concentration of H pluses in the solution. So acids and bases. This is a bit of a repeat as well. Two major fluid compartments, extracellular and intracellular. The extracellular includes interstitial fluid and a small component of blood plasma. So this is a reiteration of the previous slides. And a reminder that extracellular fluid has high levels of sodium and chloride. Intracellular fluid has high levels of potassium, phosphate, magnesium, whatever. It's yeah, okay, fine. All right. Extracellular fluid has high sodium, high chloride, yeah, a little bicarbonate in there as well. In terms of intervention and or behavior and body response, if we need to bring in fluid, okay, whether that's through body mechanisms or through therapies, like for instance, severe dehydration, we're going to have a terrible thirst response, all right, seeking fluids, kidneys are going to play a role, but we're also going to do this therapeutically. Burns, same story. We're going to be replacing fluid. Hemorrhage, all right, if you remember what I talked about with gunshot wounds. people that are slowly dying of hemorrhage from a gunshot wound. And that's the common reason people die in gunshots is that they will often experience extreme sensation of thirst. Sepsis, that's a blood infection that will lead to loss of fluid from blood plasma and edema, and any type of shock that occurs where you have a precipitous drop in blood pressure. So what do? We replace fluid volume therapeutically in order to maintain blood flow and perfusion and nutrition and oxygenation. Any type of intervention needs to be sensitive to the patient's situation. That leads to more complex diagnosis and therapeutic questions. But if we have low fluid volume, we're going to do something that called fluid challenge, which you just can't just give them water. That will lead to water toxicity. So we have to add what we call osmotic solutions, and that can be normal saline, 0.9%, sodium chloride. That's by far the most common. It's isoosmotic. Only people that are already hypertensive would not be indicated for normal saline. Lactated ringers, that's another, it's got lactic acid in it and some other molecules. A little more of a nuanced situation. This one is often used in people that have chest pain and so forth. And we can do a D5 as well in relatively specialized situations where a patient might be experiencing hypoglycemia, etc. Just as an example of why we need to pay attention to this is that, we are in an environment where people are frequently consuming water and or electrolyte solutions in classes. It's more common than not now. I've seen the number of drinking bottles, increase exponentially in the last 5 to 10 years. It used to be relatively uncommon, and now it's the majority. But if you take in too much water too quickly, you can and will die. This gal here was in a contest for a gaming system in the early 2000s, and they had a water drinking contest, and she ended up dying hours after this. Careers were ended, lawsuits, and so forth. Definitely don't want to go there. In a perfect world, we would see a balance between intake and output. Now, remember that not all fluid intake is drinking. There is a substantial amount of water in food, all right, especially food that isn't very salty, like, pizza doesn't have a lot of water in it, but fresh vegetables, all right, soups generally have a fair amount of water in them. So our average intake, in most situations is around 2.5 liters every 24 hours. Mainly driven by a thirst response because it's going to affect our behaviors. It's hard for us to metabolically generate more water if we need it, so we need to use complex behavioral patterns because we are animals. If we were plants, what we would do is we would have a genetic response in order to absorb more water from the soil. Same thing with output. So intake and output should be balanced in a typical homeostatic situation. Lose a fair amount by the kidneys, but we also evaporate. We also breathe off. That's particularly if we are in drier, higher altitude conditions. We call that insensible loss. Okay. We can sweat. We can also remove fluids from the body. We call those sensible losses. When we can see the fluid leaving us. If we have excess loss relative to intake, dehydration, excess intake or reduced loss can lead to edema. So if you have kidney failure, along those lines, you would see edema because you're not eliminating water properly. And we've seen this before. So extracellular fluid, what do we have? We've got sodium and chloride and a certain degree of bicarbonate. along with water in the extracellular fluid compartments. Yes, there are lots of other things, but these are the major ones. So we can replace extracellular fluid most commonly with normal saline. Right? It's an isoosmotic situation, also called isotonic. Isoosmotic tells you the constituency of the fluid. The tonic tells you the impact, and there are some variations, and again, advanced physiology. So normal saline is isotonic as well as isoosmotic, and so the cells are just fine. They're going to lose water as fast as they gain it, and so there's no change. If we have too much solute, we lose water. This is crenation. All right, this is the crenation response that we've been talking about. And if we have too much water but not enough solute outside the cell, then we get cell swelling, and in the case of red blood cells, rupture. Now, red blood cells are a little bit of an acute situation because red blood cells have water channels, and so they equilibrate extremely rapidly. I mean, literally seconds. And so they are not as good of a representative of all the tissues in our body as we'd like to, but certainly conceptually, red blood cells make a wonderful acute example of crenation and edema. Again, a reiteration of what do we see inside cells, intracellular. Certainly the majority of water and fluid are inside cells to the tune of 2/3. Potassium is the big one. Phosphate's the next biggest one, and a distant third place is magnesium. But potassium's a huge one. Phosphate's in there, of course, and a little bit of magnesium. And the reiteration goes on. Sodium is the major extracellular ion. It's cation, and remember that the T is like a plus, so that means it's got a positive charge. Certainly important for neuromuscular function and so to maintain proper fluid balance. It's the primary driver of fluid retention or fluid loss in the kidneys, which the hormone that affects the kidneys, we had talked about anti-natriuretic peptide, but also even bigger aldosterone. And potassium is the major intracellular ion, abbreviated K for kalium. It's really, really important for that resting membrane potential that we spend an enormous number of calories every day maintaining. Certainly necessary for the excitation, part of the excitation contraction process in muscles, as well as action potentials and so forth for nerves. It's also regulated by aldosterone. In fact, many of the diuretics, the things that we use to create more urine, try to get rid of fluid from the body, have a nasty little side. It's not nasty, I shouldn't say it that way, but have a notable side effect that you will often lose potassium alongside the water and the sodium. So hypokalemia is a common side effect in diuretics. Now they do have potassium-sparing diuretics. The problem with potassium-sparing diuretics is you can get hyperkalium. All right, so it's sort of a double-edged sword sometimes. All right, so hypokalemia, that's too low blood potassium. The most common cause of low blood potassium is diuretic use or poor nutrition. Hyperkalemia, that can be due to kidney issues and or the use of what are called potassium-sparing diuretics. And in a future life, I'm probably going to shift these slides around because we've been talking about membrane potential. Well, here it is. In the extracellular compartment, we tend to have high levels of sodium and chloride. That's because we have transporters that do that. We use ATP, big chunk of it, to send sodium out. Chloride doesn't normally move around much. There are exceptions, but again, chloride is normally not involved here. On the other hand, potassium, a little bit of phosphate, some proteins in the inside. Potassium is the big gun inside the cell. And creating that gradient of sodium and potassium is what establishes the voltage change across this membrane. Another bit of a repetitive slide on calcium. Calcium is very important for enzymatic function. It's a very important signaling molecule, particularly in muscles, cardiac and skeletal muscle. It's important as a cofactor for enzymatic, primarily regulated by this hormone called the parathyroid hormone. Magnesium, while a distant, smaller influence overall, is still a big one for muscle contraction, heart contraction, and neurologic function in a more specialized way. So we regulate it quite significantly. Chloride, it follows sodium most often. It's an extracellular anion. It is not the chief one. It is one. But sodium is really the big gun here. In respiratory and digestive situations, chloride has a bigger importance, but we're not going to worry about that on a general sense. So some quick functions of calcium. It's involved in the mineralization of bones. So if you have osteoporosis, what's normally happening there is that you might be hypercalcemic because you're breaking the bones down, and now the calcium's liberated out into the blood. You could also get osteoporosis from hypocalcemia, so the body's trying to compensate. Teeth, it's involved in the ceramic and the dentin. It's certainly the second message in muscle contraction, that you have an action potential that then causes liberation of calcium inside muscle cells that then causes contraction. Nerve muscle conduction, primarily where it's going to be involved in the release of neurotransmitter. So when we get to the presynaptic terminal, and we're going to send a signal to another neuron or a gland or a muscle, you use calcium in order to release that neurotransmitter. And again, enzymatic function, blood clotting requires calcium. And a quick definition for you: if you experience caliuresis, that means that you are losing a lot of potassium in the urine, that your urine potassium levels are clinically high. And again, we're not going to worry about this, but the most common reason that you will see day to day in the clinic caliuresis is due to diuretic use. People have high blood pressure, and so they're using diuretics, and then the side effect of the diuretics is caliuresis. Most often we treat that by just giving them some potassium supplement. With bicarbonate, again, we've talked about this before, it is extracellular. It's very much involved in acid-base regulation because we can use that formula that you saw earlier in order to transport CO2. About 2/3, maybe as much as 3/4 of carbon dioxide is carried in the form of bicarbonate. It is a base. It will accept protons, so it's a mild buffer. It will give off protons if needed as well. And another summary slide for you with regard to pH. This is the concentration of protons. Again, a lower pH is acidic. A higher pH is basic, ranges from 0 to 14. We really only ever see 3.5 to 4.5 in the 7 range, 7.35, 7.45. Anything out that the body's going to need to respond and/or therapy. So anything below 7.35 is clinical acidosis. Anything higher than 7.5 is clinical alkalosis. Pragmatically speaking, alkalosis is a lot harder to treat than acidosis is. A common cause of alkalosis is transient, when you hyperventilate, like when you laugh and you get hiccups. The hiccups is a spasm of the diaphragm in response to hyperventilation, loss of CO2, alkalosis. So you interrupt the breathing pattern, you get hiccups, and that hopefully returns you back to a normal pH. And we have chemical ways of regulating pH. We have buffers. We talked about phosphate. We talked about bicarbonate is the big one. is the really big one. Phosphates in there. Amino acids and proteins are a distant third. You can regulate your breathing patterns by depth and frequency, because if you either retain CO2 by reducing your depth and frequency, you can become more acidotic, or you can become more alkalotic by increasing depth and frequency in order to breathe off CO2. If you've ever seen somebody in a panic attack, one of the things they might have them do is breathe into a paper bag, because when you have a panic attack, you hyperventilate, become alkalotic. So what do? You breathe into a paper bag and you're recycling that air, increasing CO2. And then we have the organ for this is good for volatile acids, not fixed acids. We can either directly excrete protons, or we can resorb or create bicarbonate. Again, in the earlier slides, it was much more in depth. This is a good summary slide for us. And so another review slide on what acidosis is. I won't belabor this too much. What the key point would be this last right here. What are the major causes of acidosis? If we have non-respiratory, so one acidosis would be heart failure or choking and not breathing. That's what we call respiratory acidosis. But if we have kidney disease, issues with diabetes, or severe diarrhea, we're going to be losing a lot of fluid and or electrolytes. You can also have, again, extended periods of time of ischemia where you build up lactic acid. And here's the primary way with how ventilation affects pH. This is a great formula to memorize. All right, so, and I'm going to start over here on the right, because CO2, if you blow it out or that sort of a thing, you build up of CO2, that reacts with water to form carbonic acid. We actually have enzymes that will facilitate this. And then carbonic acid in physiologic pH will usually release at least one proton and form bicarbonate, or bicarb is what they sometimes talk about it. All right. So by changing the rate and breadth of breathing, depth of breathing, we can regulate this, and the mass will move one side or another depending on how much CO2 we have. Then we have alkalosis, which means we're above 7.45. We can get this through hyperventilation, like for instance, panic attacks. All right, we don't have enough CO2, so we have a rise in pH. Or fixed acids, persistent vomiting, because we are pumping protons into stomach acid directly. If you're vomiting that out, you're losing acid that would normally be resorbed. So that's a metabolic alkalosis. Persistent vomiting, severe vomiting, can cause metabolic alkalosis. Another situation of metabolic alkalosis is taking too many antacids for ulcers or gastric irritation. The remainder of the slides have some quick questions. Great to ask yourself, great to ask other people to get you on the messages. So how do we regulate blood pH? Well, in situ, in the extracellular compartment, immediately we have buffers, and bicarbonate's the biggest one. Secondarily, also relatively quick, is the respiratory system, by how fast and how deeply we breathe. And then the slowest, but arguably the most powerful, is the kidneys, either through management of protons into the filtrate or resorbing and creating bicarbonate. What is another cause of acidosis besides respiratory? Can be due to the failure of the other control systems, kidney in particular, or diarrhea, or buildup of lactic acid. The most common thing that I can think of with buildup of lactic acid would be hypermetabolic syndromes, which again get beyond our. You have serotonin syndrome. That's one that I'm thinking of. A response to certain types of anesthetics can do this. But the one that I think of as a rock climber and a caver and somebody who spends a lot of time on rope, this one can be triggered by what we call a harness hang. When somebody, especially somebody who's unconscious, is hanging in a harness, that harness acts as a mild tourniquet, and prevents venous return from especially the lower legs and the lower appendages. And if they've been hanging for a long period of time, that lactic acid builds up, and suddenly they're brought off rope or off that hang, and all of those components rush into the rest of the blood compartment and lead to a metabolic acidosis. So complicated. Try to keep it simple. Try to keep it simple. Is it breathing? All right. Or is it kidneys? Or diet and things along those lines. It's simple messages, not super detailed, complex messages. Again, the two most common causes of metabolic, again, fixed acid alkalosis would be taking too many antacids. All right. Again, people that have either undiagnosed or are not seeking treatment for ulcers, they might just try to do an over-the-counter remedy because of expense, and then so they over-ingest antacids. Or vomiting is the single most common metabolic alkalosis that we see. Kidneys can also play a role here if they're not regulating bicarbonate correctly. And so that's it. That's it. So a lot of this second lecture is, second part is a lot of it's reiterating what we already saw in the earlier part of the lecture. This is a bit to reinforce the lessons that we learned early on in the lecture, to try to get distilled down to digestible concepts and messages. As always, bring your questions. Let's talk about this stuff, and I look forward to seeing you in class. Thanks for your time.